I welcome everyone to the Tools and Technology Seminar Series. For any of you who have not been here before, this is a venue to discuss new tools, technologies, methodologies, either recently developed, they could still be in development, they don't actually have to be a finished product yet. It's a great opportunity to discuss things you're working on, but also to discuss finished tools and also just tools that are of interest to researchers and maybe you found useful in your work and you think others may also find useful. I guess I should introduce myself. I'm Marcy Brandenburg. I'm the bioinformationist with the Tubman Health Sciences Library and also with DCMB. I do the programming for the seminar series, so if you're interested in giving a talk next semester, please let me know. I'm always looking for new speakers. There's a sign-in sheet that's going around. Please sign it. It helps us justify the pizza, so if you want to continue having pizza during the seminar series, please make sure you sign in today and in future weeks. So I think that's all I have to say logistically for the seminar series. So to introduce our speaker, I think Indika will just say a few words.
Yeah, I'm good. Okay. So Haiming is in my lab for almost 2 years now. A wonderful colleague, and he's a senior fellow. So Haiming, I want to make a few comments. So Haiming is the first guy I will say maybe in the world who reprogrammed a bipolar patient's skin cell to a neuron. So that's how I met Haiming, and now he's doing wonderful things in my lab. So today he will present his work. Yeah, he used to work with Melvin. Yeah. Haiming, you want to?
You guys own.
Okay, good afternoon. Thanks for the introduction, and I'm happy to be here today. Actually, I'm fortunate to have the opportunity to work with Indiga and taking my interest in research in a new area. So today's topic is going to be chromosome capture to study the genome. So the genome we're talking about here is the human genome. I put a 3D here. I'm focusing on the cell in the nucleus. That's the 3D shape. But Indica's lab is a 4D lab. So we, in addition to study the structure of the 3D cell, we add how the cells behave or how the cells function over time. Okay, that's the 4D lab. Okay, now let me begin with a relatively familiar knowledge about what we know in the nucleus. That's the introduction. Then my second part of the talk will be the real experiments data we generated and published or to be published soon. And finally, I'm going to end with the talk with the starting of a new project that we're going to be doing more exciting experiments. Now let me begin with the introduction. So this is a diagram that I'm taking from this paper from this Professor Rudin at the Fred Hutch. The diagram summarizes what we know in the nucleus regarding talk about the human genome or the inheritance information in the genome. The top line is a double helix. That's known. That's solved in 1953. Okay. This is known for centuries, the chromosomes. In between of these two well-known structures, there are existing structures we observed, which has been observed or known, or some of the structures at the first layer of higher structure of the human genome is the so-called beads on string. Okay, so the beads are the octamer of 8 histones, the core, and the string is the double helix. That double helix is super coiled on the octamer histones, which is 1.6710. In between each bead, there is about 10 to 80 base pairs of link. This structure, I think, is well known now, well accepted. But between this and this, these are the structures we know it existing, but not quite know the function. And this is a well-defined 30 nanometer fiber, which is coming from the more compact from this beads on string structure. After that, we don't quite know these things. So these are the structures actually we can see in the interface nucleus. So all these structures existing, and because of, we know the traditional story that a cell has a potential to divide into two, reproduce itself through a cell cycle, G1S, G2M phase. So between the, so from the, well, there is a G0 phase. That means the cell is quiescent. They don't do dividing, they just stay there. Once there's a need, a cell has the potential to go through this cycle to reproduce itself. In the interface, before the M phase, the structures I talked about in the first slides actually existing. And in the human genome, there are 46 chromosomes. These chromosomes in interface existing actually occupies relatively areas, or a term is given as chromosome territories. So each chromosome occupies a small region, or a region that it occupies. We don't know why it occupies that region, but it's there. And no matter what the cell goes, the cell will divide. The cell will carry these territories to the next generation. If you use a technique called in situ hybridization with color labeling, with different color labeling different chromosomes, this is how the picture looks like. So each of the color represents one pair of chromosomes. The same color represents one pair of the chromosomes. So we're interested in this interface nucleus, and we're interested in the previous slide talking about structures, folding, and any interactions between of them. So how do we do that? That's a new technique. We're trying to, well, this is a simplified diagram that represents the previous slide, the interface nucleus. Suppose the chromosomes, the chromatins, are folded in certain ways, and we try to look specifically any different chromatin, if they interact each other. What is the meaning of the interaction? So these are the questions we want to ask. Now, how do we get those things? And this is a new technique called the chromosome conformation capture. Go through well, the experiment in the lab. You grow the cells, and you cross-link the cells by formaldehyde and fix the structure of those interactions, or chromatin structures, fix it. So that's how you preserve the interactions. Then you find a way to study the preserved interactions. First of all, you cut your whole chromosome with an enzyme. We in the lab use HindIII. That's a 6 base pair sequence recognition endonuclease. So it has its specific sequence recognition ACTT. So every of these sites in the genome, it will cut. So you produce fragmented DNA. And label the end of the cutting site with a chemical biotin. Then you can reconnect the cut site with the ligase. What we need is the interactions between any two fragments at the cutting site. Those things we can get it. I mean, in our case, we're only interested in these interactions between any two fragments naturally existing. So then you pull down this and get rid of the things around. Then finally you got a library. You got a library that contains, depending on how many cells you use, billions of different molecules that has two different chromatin ligate each other from two different loci in the genome. might get billions of these, or you can get more of these. Okay, question. Well, if there's a restriction size here, of course you like that too. No, you just, if it's close enough, then you, in this case, you may form a loop. But eventually you will break it down, so you will have this part as the interaction, and this top part as another interaction.
Crosslinks do not occur at the ends.
Oh, no.
I have a question. So people have shown with 3C, which is sort of a precursor to this.
Yeah, that's
But
my.
when you fix, you distort the spatial interactions in the nucleus.
Okay.
Are you concerned about that?
Well,
sorry, it's a really loaded question. But you know what I'm saying? I'm not, but it is a possible concern, right? If you're using a formaldehyde and the nucleus swells up and distorts, then the spatial interactions, maybe especially in trans, might not be the same as they would be in the normal cell that hadn't been fixed. I don't know. This is, many histologists argue about this. Is this of any concern, or?
You may have some idea. No, absolutely. There is. We don't know. Because even imaging, you have to fix with formaldehyde.
Right.
So ideally, you have to do this in a live cell.
Right.
Not fixing, but then we have to account that. That's where I think mathematics will come to play.
We have used some. We have used some milder fixatives, methanol.
No, it's been normal. This is the standard way, and then you use again at the end to use. Again, I'm saying data is again incomplete. We use mathematics to somehow recover that disruption.
We have successfully done fish imaging with milder fixatives, but yes, it's more sensitive.
Using a cross and get the maturation.
Right. So, right.
Yeah.
Sorry.
These are big questions, but
Yeah, go ahead.
Okay, that's well. Since the talk, you mentioned the 3Cs. I mean, actually, this technique, chromosome conformation capture, starting with the 3C experiment. The 3C is just looking for specifically you're interested in two different fragments that if they interact with each other or not. That's the one-by-one interaction. And then during down the way that during the development, I mean, the maturation of this experiment is going through a 4C experiment that you are interested in a locus, how the locus interact overall in the genome. That's the 4C, and one to many interactions. And then this 5C thing, you can think it's a multi-4C project. You're interested in many loci in the genome and how the many loci interact in the genome. So, and then the highest level now is the high C that you just do the whole genome. You don't care about the 4C sequence. And you solve problems with mathematics. Okay, now, so after this, you got a map like this. This map is it's a plot from a matrix. We call it high C matrix. Okay, so.
Is one chromosome or four?
Yeah, you do chromosome by chromosome.
I mean the contact matrix. How many chromosomes are there?
Well, this is the diagram shows how the matrix look like. Yeah. Or next, let's say for example, chromosome 8 looks like this, and the phenomenon of these maps is actually diagonally dominant. So you have more interaction fragments that identified within a certain region. Well, let me give you why I should give you this idea now. How this matrix is made. A matrix, you can make a matrix at a given resolution. You divide the genome into 1 megabase equally, or 100 base equally, or 1 base or 1 KB equally. It depends on your resolution. So let's say we divide the genome into 1 megabase pieces. So each megabase is a column. And then you, based on your library, your sequencing reads, you map it back in the genome, and you stuff the fragment into one bean if it comes from the same megabase. And then that's the read in the cell you're looking for. So the whole thing is going to be like that. And naturally, I mean, not naturally, or it is if it is naturally. Let me say it in another way. If the map represents the natural way chromatin interact in the cells, this is good. That means DNA fragments close to each other, they intend to have a high rate of interactions. And they far away, they tend not to interact with each other quite frequently. Okay. That's the meaning of this. Now, this is the introduction. If I gave you clear pictures of how this map look like, that's good. Now, let's go to the data and the projects that we have published or we are going to write up.
So just a side note question. So when you have a contact, do you have a intuition about what the actual physical distance is there?
Yeah, we know the sequence. So we know where the contact comes from.
But the sequences are...
The sequences are based on the whole genome.
I'm asking about the distance between...
So he
Oh,
asked,
yeah, the distance between. Yeah. So in our case, it's defined by the restriction sites.
Which is what sort of distance?
Yeah. Physical distance by base pairs.
So
I mean, spatially, we don't know yet. Spatially. Sorry.
Do you have a feel for what that average distance is?
Of
The average
cuttings
distance?
or something like that.
You mean?
Well, let's say you do cross-linking, then you say the distance between two segments will be defined by the length of the cross-link. In this case, you're using some kind of reagent to cross-link the DNA segments. What's the distance of that physical?
Okay. I wish I know. Yeah, spatially I don't know, but at the DNA sequence level we know.
so this is, I like to think this is like some sort of coincidences in the genome. You have no idea the minimum or maximum in this medium space.
You don't have an idea of the transient nature.
So how typical is this figure of high-C data? Because what I remember seeing is more like boxes along the diagonal with some white space going close, whereas this just looks like a smear of red all the way down.
You mean these diagonal things, like, or the whole
Yeah, so
thing?
you can see the boxes more on the upper left. That's kind of more what I'm used to seeing.
Yeah, because chromosome 8, say, it has 400 genes. You have each of the square. I mean, each of the block comes from maybe several beans. You look at the heat map. This doesn't distinguish this yet, but you have a way to distinguish it.
So if we zoomed in on there, you
Yeah, let's zoom in. You can see a fine structure.
And I guess you're used to looking at normalized data because once you normalize it, we remove the strong diagonal.
And it's also low resolution. The tabs are typically smaller than.
So now the first paper, I mean, this is a simple experiment. We're interested in asking a simple question because many of the experiments was done in cells on the culture dish that grow in two-dimensional. And actually, humans or living organisms are the three structures. So we try to see if there's any differences between between two-dimensional cells, no, two-dimensional cultures, and versus the three-dimensional cultures. So that's what this paper talk about. And simple question, no further things, just the difference between the two culture conditions. That's how we got the 3D cultures. So studios, everybody knows that we grow in the dish. I mean, how many cells? The 3D, we use the techniques from this Biomatrix company. It's actually on this campus, right underneath our building. I mean, our floor in the same building. Okay, now you can use a 96 format or a 384 format and grow the cells in these droplets. it's a liquid. So the cells will form a ball or a sphere we talk about. And Scott there, he did a nice job on measuring the size or the volume of the cells that grow in two different conditions. This is the 2D growing, the 3D growing, and these are the measurements that we did. So you can see the differences between the two different conditions. So that's simply the experiment looked like. Whole genome, chromosome 1 to chromosome 22. We didn't look at chromosome X for some issues. And this is just, we do replicates on each culture and condition. So 2 replicates on each 3D and 2D. We're looking for how similar between the 2 replicates in the same condition, and how different between the 2 different conditions. So on this diagonal is the matrix, the whole genome matrix that the contact might look like. So of the diagonals, these are the matrices that you calculate the differences between the diagonal matrices. And you clearly see that there's very differences between these 2, the same replicates, or these 2 from the same replicates. But there are larger differences in terms of frequencies of interactions over the genome that between the 2 culture conditions. That's this figure talk about.
So you're saying in 2D there are more off-axis interactions than showing?
So this is a heat map, just a scale.
Yeah,
that.
So are they similar? Is one more than the other?
This is well. If you look at this, that's the difference between these 2.
Yes.
Is this.
Yes, I know.
So there are some differences, but I think it's the minimum.
It's minimal.
Yeah. And if you look at these 4 plots,
yeah,
that's this one is between these 2.
Yep.
And this one is between this one and this one.
Yep.
Okay. So you can see this all that. I mean, we're just looking for the diagonal matrices and compared with this. This is the replicates differences, and these are condition differences. So this indicates that we do see some differences. How different is this? Well, just get some measure. Before I get into that point, this is taking chromosomes, specific chromosomes. You look at, in your matrix you have beans, right? You just in each of the chromosomes, let's say you have chromosome 1, you have 250 beans. You sum up the beans and you make the plot. This is just a total width in each of the columns. Chromosome 1, 6, 11, 14 tend to be the colors. Let me indicate. Green and blue color are the 3D culture replicates. Okay. And the pink and the red lines are the 2D culture replicates. Okay. So you can see that between the replicates, they tend to be similar counts in each of the beans or each of the columns of the matrix. But between the 2 different conditions, the counts or the total counts at each column of the matrix, you can see some huge differences. But in some of the cases, let's say chromosome 19, chromosome 22, tend to has less counts in 3D cultures. But in some other chromosomes, actually, 3D cultures has more counts. So this difference is
there's a certain number of mega.
Yeah, let's say we define. Yeah, in this case, we define as 1 megabase sequence.
But it can be defined.
Because chromosome 1 has like 4,000 genes.
Well,
22 has maybe.
yeah, we're not defined by genes. We just take the sequence. Let's say chromosome 1, you have 220 megabases. Then the chromosome 1 matrix will be 220 by 220.
And certain features can be better resolved by using bins that take genes into account.
Okay. Now we do calculate some statistics using techniques, and the differences we saw from the previous slide actually can be shown in this heat map. Blue colors represent significance. These are actually good, and it matches the previous slides.
Blue represents what? Significant differences?
The differences between 2D and 3D conditions. Yeah. Okay, now that's our first small project. Give you a primer how a big project may be performable or operatable. Now this is the project that in our PNS paper recently, and these two guys, the double chain, and equally, and he did a lot of work on analysis, computation, and he's back in China now, and he took a full faculty job in one of the big universities in the city. You know, if you have been went in China, Xi'an is the, you know, that's the very old city, right? Yeah. Okay. I have to acknowledge him quite a lot, and because he's not here. Now, the project is supported by DARPA and motivated by the question of biochronicity, actually biological clock. And we want to take a simple experiment that answer a complex question. But in this simple experiment, we cannot answer all the questions. We may be able to answer some of the questions. How the interactions, we gave them a name, form. That's Indica's term, form. And how form link to gene expression, we call it function. So how form and function relate to each other and over time. So that's what a 3D genome plus the time, that's a 4D genome. So this is the scheme of the experiment. We capture function by reading RNA molecules, and we capture form by this Hi-C technique. Our time, our 56 hours, every 8 hours, we sampling the cells for Hi-C, and every 4 hours, we sampling the cells for RNA-seq and fish. So that's how it goes.
Light and condition.
Sorry?
The light and the darkness condition.
Light and darkness conditions. Normal culture room with the incubator at 37 degrees, 5% CO2. Light is over the night. Is whole time is dark unless you take the cells out.
What is the meaning of P2 Cry1? They are all blue light sensing pathways. And why do you want to study this for if you keep it in the same darkness condition or light condition?
That's a good question. Well, in my limited knowledge, these 4 genes we're interested in because they are major master clock, biological clock genes. It functions well in the whole body based on the day, 24 hours of the expression changes. And some other people did experiments, take the cells out from your body, grow it in dish. The expression pattern of these genes keeps the same.
When you synchronize.
When you synchronize, yeah.
Yeah.
So that's, yeah, your question is good.
You haven't looked at glucocorticoid receptor or any other nuclear receptors?
We have RNA-seq data. We not specifically looked, but we have those tables, analysis of RNA-seq data, the tables that in the supplemental documentary in the PNS paper.
Okay.
You can check off any of the genes.
These things are all highly regulated by, or regulated, right?
We have a few more papers coming on this.
Okay.
So, this is the thing I learned from Indiga, and he teach me heavy, and spend a lot of time. Okay. One of the ways to look at the interaction map or the contact map, the Hi-C map, if you will, is using this Laplacian approach. So you construct a Laplacian from your Hi-C map. So that's the formula, Laplacian. After you get your Laplacian, that's a matrix. Okay? Then from that matrix, you get eigenvalues and eigenvectors. Okay? These eigenvalues, you use it as a quantitative or relative quantitative description of the stability under the structure stability. And eigenvectors, you can use it as a partitioning vector that you can partition the genome into different domains. So that's how these two vectors. Well, specifically, not all the eigenvectors are used. We specifically in this case, the smallest eigenvalues and eigenvectors we use.
Why?
Small.
That's the name of the mathematician called Federer. He solved this.
And how did you do it? Was it?
I mean, I think
mathematical foundation, or is this phenomenological?
No, it has a geometric foundation.
Okay. I just don't know.
Fine.
Are the other ones? The second smallest eigenvalue of the Laplacian is called algebraic connectivity. That will tell how. So it's good. Yeah, we can talk later.
Yeah, no, that's good.
Okay, so this is the soul of the paper, and we just get the information from the Hi-C matrix, and we look for the 4D patterns. One of the figure from the paper, and that's the main part of that paper. So we have the Hi-C matrix. For example, this is given by chromosome 4. Okay. This so we have the information here, RNA-seq data, and second this eigenvector. Okay, Hi-C matrix. So using this eigenvector, you can see this chromosome is partitioned into two domains. This actually is well accepted now from the very first Hi-C paper. They use a different approach, mathematical approach, and we use the eigenvector here. We ended up with the same, pretty much the same partitioning of any of the chromosomes. So based on the activity of this gene expression in each of the beans, we can relatively identify each of these partitioning domains with either active or not active or mixed. So this is, let me take you through the middle part first. So in this Hi-C map, there's another term here I have to introduce to you. You see these blocks? Different colors, right? These blocks, squares. These are called topologically associating domains, which is first described in 2012 by Bing Wen's group at UCSD. So this is now a concept in this field. Okay, the genomes are organized into tags. Okay, each tag actually is a partition of a domain that you can see it by the eigenvectors. So specifically, if you're taking one tag, for example, this one, it has 4 genes in it. The transcription activity of these 4 genes actually is 0. So this is called an inactive tag. Well, this middle one is active, and genes in this tag, the 3 genes, they tend to be, the expression pattern of these genes tend to be synchronized. And this tag has 6 genes. Some of the genes are inactive. Some of the genes are expressed but not quite well synchronized. So that's the way we describe this tag's activity in terms of form and function: inactive, active, mixed. So if I explain clearly this picture to you, then the paper is good.
So I have another question. I'm sorry, I didn't ask one. Have you thought about corticosteroid or cortisol treated and untreated?
Yes. And one point. When we do the experiment, the cells are growing. We synchronize the cells with the cell cycle by just serum starvation. Right? In the meanwhile, we synchronize the clock by glucocorticoids, by dexamethasone. It's a well-known chemical to synchronize the clock. The clock, or the expression of the clock genes, actually.
So dexamethasone, which is a synthetic, wasn't there, what would happen? They would just become unsynchronized. And it'd be all over the place. Or?
We have the data.
I think
the clock genes themselves would not be synchronized.
Right, I know that.
But I don't. They're not in the same tag, though, so I would guess that the rest of the tag might still. We could look at it.
Well, only because there's a normal cortisol rhythm in humans, right? That's high in the morning, and anyway, it has a specific curve, and it's flattened in many stress-related disorders, like psychiatric disorders.
Yeah.
And so, I'm just trying to think of an experimental way to look at the clock for cryptochrome genes in that manner. That makes sense to you. Mm-hmm. But anyway, go ahead. I'm sorry.
Some data we never. A lot of data still to be analyzed.
Be very interesting.
Well, you gotta. I mean, that's a good point, but I have to explain to you one more time. One thing, when we synchronize the clock, dexamethasone is added at one time and washed away.
Yep, I understand that.
That's it. No simulation with the human body.
Right, you're not adding it or having different times.
So just to make sure I'm missing something. So your topological blocks, you had a name for it, that comes from your analysis.
That comes from.
This comes from your
the analysis and defined by some criteria.
Is that predictive of your panel on the right?
The blocks predictive.
The blocks then are synced, is what you're saying. You can predict that from the structure that you get from
your. You mean active, inactive, and. Okay, good question, Indika.
So you get the domain structure, the topological structure from your eigen composition, right?
Sure,
But then your panel on your right, where you're selecting a couple of these blocks and looking at.
So those are the blocks read, and those are the genes.
Yes. So the genes within a block.
Yeah.
You find the block from your analysis.
So that
genes
big block,
is
say
this
D1, it's a one block gene diagram.
Right.
That is the pad, if you will.
Yes.
The D1.
Yes. But then the last panel on the right, for example, where they're mixed, you're saying.
Yeah.
Is that predictive from the block structure that you find? Can you tell whether
from
they're mixed
data.
or synchronized?
There's no prediction from there.
So what's the purpose then of the?
So see that from structure.
Well, maybe beyond our present knowledge.
No. So they are maybe structurally organized, but then the activities are not.
Yeah, it's not necessarily functional.
So
functionally.
But the structural organization isn't coincident with their activity.
No, because there must be period in other genes and other paths, right?
Yeah.
And also.
Because still we don't know the mapping between function, structure, and function.
Right. That's what I'm saying. It's still to be discovered.
But then, and everything's along the diagonal that you're looking at here, so there's no far from the diagonal blocks, right? There's no distal sites in the genome that are really in a topological.
Distal, but these are big tags. These belong.
Yeah, they're things out laterally, right, off the axis.
Not related to that. You do see, when you divide it into the
structure is
two
really
sections, you do see that generally
critical
one is all active and one is not. So at that level, so when you divide it into just the two rather than looking at individual ones, you tend to see that one of them tends to have all the like active regions, the other tends to have all the non-active. So in that way, you can kind of predict it, but it's not.
But you didn't learn anything more than just knowing they were sequential.
Yeah, but
Those genes
this
are
is
sequential, so
this is
they're gonna
great fertile ground for further research, because maybe the genes in the clock pad have something that do, well, of course, I believe structure and function are intimate.
Absolutely.
So this is
absolutely.
for further discovery,
Yeah,
right?
absolutely. Yeah, because again, I tend to think it's like fibroblast cells are not a differentiated cell type. So that has some intrinsic noise to fibroblasts.
Yes.
So I guess the missing information here you didn't see is, because this is from one time point. We have 8 time points. You would, if you plot this into, if we put all 8 time points, you have to see 8 of these goes on. So one of the information. And the other information here is the TADs. Well, now it's well accepted. TADs are preserved in the genome, cell type invariant, and preserved between vertebrates. Of course, there are changes in TAD structure when the cells are different or when there is some interruptions or perturbations. Okay. So
I have a general question. For all these kinds of studies, were there any features in this system that you were able to make predictions before you did the experiment? So the experimental results would test certain predictions pushed onto other things as well. That's the difference between a description of what was done and what was found and a testing of a hypothesis or a testing of a prediction. Is it too early to be doing that in this system?
I think it's too early.
But
It's hard because you don't know the right answer for the chromosome structure. Like there's no golden standard to compare. You know, it's not like we have a mapping of a chromosome structure to compare.
so we need.
I agree.
But
So we have few
there's a hypothesis.
Yeah, somewhat. Laura has a paper now. This cancer, we know some.
Yeah, the cancer gives you a little bit more testable.
Yeah, and then we have another paper for the same data. We have a different approach to looking at this, the barriers. So yeah, so to be seen. So I can talk some research.
For example, there are time course data on transcript expression. And there are transcripts that lead to protein-protein interactions. So those we know quite a lot about, whether it's for cancers or for other normal physiology, biochemical physiology.
So if we move on, that's another thing we said or we talked about in the paper. This is actually Indica formulated this, and we talk about networks or gene-gene interactions or loci-loci interactions in that paper. This is the slide that helps us to understand what the networks were meaning in that paper. Given 2 genes, gene 1, gene 2, we have interaction, high-seq data, we have gene expression data, and we could calculate the so-called feeder number on this. So these are the 3 conditions that we're looking for in order for us to say these 2 genes actually interact with each other, or you can put an edge between these 2 nodes. Right. So another condition here is that if the genes whether the genes has or have common transcription factors. So if gene 1, gene 2 has gene 1 or gene 2 has very specific transcription factor binding sites on both, it's good. And we look for these parameters, and then we relatively say these 2 genes tend to interact with each other. Whether they are in the space of another, well, it's too large, but transcription factory. So that was another term that described for a long time transcription factors. Different genes from different chromosome or chromosome loci go into a hub. That hub may have, the transcription activity. So the genes has to go there and make the transcripts in the meanwhile. So this also answered that question. So that's what I'm going to end with for this paper. Now, no fix for this paper. We are writing the paper. Laura is taking the reading level and doing all this computation work and analysis and she will give a talk in December, right? So you will see that more details in there. But this cancer cell is a cell line from a colorectal cancer patient, a well-studied cell line called HT29, which has, it's an annual protein. It has about 67 to 70 chromosomes in the cell, has different type of translocations and different types of insertions or deletions or chromosome duplications or regional large segments of amplifications, specifically in the MYC region. Okay, that's phenomenal. Okay, now time is okay. And with the experiments, now it's time to start a new experiment. That's what we're going to look, what we're going to do. MyoD is a myogenic differentiation gene in the genome. It's well known that it's a master transcription factor that one gene makes a different cell into a muscle cell. But the details, they say how this gene works and if the gene changes the structure of the genome or reorganizes the genome or how the gene functions during the time that it changes the cell into another, we call reprogramming state. So now in this case, we try to reprogram a human fibroblast cell line into a myotube. So that's the experiment we're going to do. Just using two approaches. One, trans-express a gene, the myodi, and look for changes or sampling data. And the other thing, using a new technique called the CRISPR-Cas9 that specifically activate the myodi gene in the cell because the myodi is not expressed in fibroblast. So we'll see what's going to happen in, well, hopefully a short period of time, but how fast we can go, we don't know yet. Ideally, this is Indica's goal, perhaps, or ultimate goal. That's a so. Given you want to have a cell type that you want to, and you understand fully the reprogramming programs, and you identify master regulators or transcription factors. I want a heart cell from a fibroblast. Which are the transcription factors we can use effectively turn that into? And I mean, this is known. So in hematopoietic development, and red blood cell from a cell, a stem cell has a nucleus, and at the end of the differentiation, the red cell has lost its nucleus. And one phenomenon of this is that some of the genes or locals that changes the localization in the nucleus from the peripheral into the center of the nucleus. So this is observed, and this is well known. But now these stories are known from fibroblasts. You can get a IPS by 4 factors, Yamanaka factors were called, and I've done this before, and we made neurons from bipolar patients to neurons. And now that's what we're going to try to answer the question here. Fibroblast to myotube with this specific one very clean transcription factor. Okay, And many people were involved, and I've mentioned the lab, everybody in the lab, and Walter is, well, tell me the story, Walter and me, when we're sampling. Sleep in the lab for 3 days, and overnight, and then go home. And every 2 hours, you take a nap about 15 minutes, and you keep on going 3 days. Right. That's okay, so this is supported by DARPA and one from the NIH Liga. Any questions?
Whole new system to do all kinds of experiments.
Is it possible to construct the chromosome structure out of your container? No.
Chromosome structure out of the.
Many attempts have been made with variable, but at best incomplete.
Yeah.
There are some huge difficulties that haven't been resolved.
Yeah. My answer would tend to be no, because you have 2 copies of each chromosome and you can't tell what's from what. And so at the very least, you're trying to do some average of the two. And then you're averaging.
You can do haplo-Hi-C. You can do Hi-C in monoploid organisms.
Yeah, in a monoploid.
The mitochondrial genome
Chromosomes
not
within male cells. That's a few ways around that.
Yeah, there's a couple.
It's very good. And even forgetting about that, the contact maps inherently don't contain long distance maps.
Yeah.
And they're ensemble.
Yeah.